All right, so let's continue with chapter 12, protections of the brain. Brain protection, there's basically three features that play a role in protecting the brain. Once you get inside the skull, of course, protects it externally. These are the things that protect the brain internally. You have the cranial meninges. There's the three layers of the membranes which surround the brain. You have the cerebrospinal fluid, which is the fluid that bathes the brain, fills the cavity, also plays a role in cushioning. And then the blood-brain barrier that prevents substances from entering the brain and its cells from the blood. This is what prevents lots of infections and those kinds of things from crossing into the tissue of the brain. The meninges are composed of three protective membrane layers. They are composed of dense, irregular collagenous tissue. Structurally, they are arranged from superficial, which would be the most external component, to deep. There is the epidural space. Then you have what is referred to as the dura mater. Make sure you notice that word is mater, M-A-T-E-R, not matter, M-A-T-T-E-R. So let's don't confuse white and gray matter with the dura mater, the arachnoid mater, and the pia mater. Underneath the dura mater, you have the subdural space. Then you have the arachnoid mater. Arachnoid, for those of you who don't know, arachnids, arachnoid refers to things like spiders. And the arachnoid mater kind of looks like a spider's web. You'll see it on a drawing here in just a moment. Then beneath the arachnoid mater, there is the subarachnoid space. And then the very interior membrane is the pia mater. So the epidural space, again, the area between the surface of the cranial bones and the outer surface of the dura mater. This is the potential space. It's only a potential space, as the dura is normally tightly bound to the bone, only allows passage of blood vessels, and we'll see a drawing of this in just a moment. The dura mater, again, epi refers to the outside, so to the outside of the dura. The dura mater, the outermost of the meninges, is the thickest, most durable of the three meningeal layers. It is a double-layered membrane composed primarily of collagen fibers with a few elastic fibers. Either when you did or when you do your brain dissections, I understand that Mr. Porter plans on doing his brain dissections today. It's Monday where I am. You will see that encasing the brain, the big thick layer that you've got to peel off to get to the brain on your sheep's brain, I believe is what we use currently, that is the dura mater. The dura is, again, two layers. They're mostly fused together, creating a single inelastic membrane, except in a few regions, places where the dural sinuses are present. The periosteal, the outer layer, is attached to the inner surface of the bone of the cranial cavity, functions as the periosteum with an extensive blood supply in the epidural space. And then the meningeal layer, which is the inner layer, it is avascular. I'll give you a second to try to remember what avascular means. Avascular means it does not have blood vessels, and it is found superficial to the arachnoid mater. The dural sinuses are venous channels. These are channels where blood vessels can pass. These drain the cerebrospinal fluid and deoxygenated blood from the brain's very, very extensive network of veins. It is also where the meningeal dura folds over itself and courses in between the structures of the brain. And again, we'll see this on some drawings in just a moment. The dural folds include some very prominent structures. The falx cerebrae, found within the longitudinal fissure. This is what separates the left and right cerebral hemispheres. The superficial sagittal sinus, it is superficial to the falx cerebrae. The tentorium cerebelli is a partition between the cerebellum and the occipital bone, the occipital lobe of the cerebrum. Again, tentorium, you get the idea of it's sort of a tent, so it tents over the cerebrum. The falx cerebelli is a partition between the left and right hemispheres of the cerebellum. The subdural space is the serous fluid-filled space found below the dura mater but above the arachnoid mater. Again, this is where we find veins that drain blood from the brain. The arachnoid mater is the second of the meningeal layers. It lies deep to the subdural space. It is a thin web-like membrane. That's where it gets its name. It is composed primarily of dense, irregular collagenous tissue and some degree of elasticity. The little bit of movement the brain is allowed due to the arachnoid mater. And that movement is very little. The arachnoid mater is made up of the arachnoid trabeculae. These are the collagen fiber bundles and fibroblasts that anchor the arachnoid mater to the pia mater. And then the arachnoid granulations, which are called villi. These are small bundles of arachnoid tissue that project superficially, that means toward the outside, through the meningeal dura into the dural sinuses. Again, this allows for return of the cerebrospinal fluid to the bloodstream. The subarachnoid space is found deep to the arachnoid mater and superficial to the pia mater. It contains major blood vessels of the brain, and it is filled with cerebrospinal fluid. And finally, the third layer is the pia mater. It's the deepest of the meningeal layers, the only meningeal layer that has physical contact with brain tissue. It follows the contours of the brain, covering every sulcus and every fissure. It's permeable to substances in the brain, extracellular fluid, and the cerebrospinal fluid. It allows for substances to move between those fluid-filled compartments, and it helps to balance the concentration of different solutes found in those two fluids. And there is a cross-section showing all of the different meninges. So here's the skull. Next, you will find the dura mater, then the arachnoid mater. There's the blood vessels flowing in the subarachnoid space. There's the pia mater, which you can see is folded back to show that these blood vessels are going over the pia mater. There is the dural sinus, the falx cerebri, and the picture of the skull again. The dural folds and dural sinuses, here are the dural sinuses, just represented by these little sort of purplish colored areas. There's the superior sagittal sinus. There's the falx cerebrae, which is going to separate the two cerebellar, cerebral, hemispheres. There's the tentorium cerebelli, again separates the cerebellum from the cerebrum. More blown up or magnified view, here's the skull. Again, here's the superior sagittal sinus with the arachnoid granulations flowing in. This is the arachnoid mater. See, again, I told you it looked kind of like a spider web. Vein, artery. There's the cerebral gray matter, cerebral white matter. There's the falx cerebrae coming down. There's the dural sinus. Subdural space here again, not filled with fluid. It's just a place for blood vessels to transfer. And then there's the arachnoid mater, subarachnoid space, and then this little line here is the pia mater. The ventricles, we mentioned the ventricles in a previous lecture. These are four-linked cavities within the brain. They are continuous with the spinal cord's central canal. They are lined with epidermal cells, and it is filled with cerebrospinal fluid. The right and left ventricles, they used to be called the first and second ventricles. Some textbooks will still refer to them as the first and second ventricle. The right and left simply means that one's on the right, in the right cerebral hemisphere, and the other one's in the left. They resemble ram's horns when observed in an anterior view, in other words, looking at it from the front. If you're looking at it from the side, they look sort of horseshoe shaped. There are three regions of each one. There's the anterior horn, the inferior horn, and the posterior horn. If you want to get a good look at what these look like, ask your lab instructor to let you see the model of the brain ventricles. The third ventricle is a narrow cavity between the two lobes of the diencephalon. It is connected to the lateral ventricles by the interventricular foramen. The fourth ventricle is found between the pons and the cerebellum. It is connected to the third ventricle by the cerebral aqueduct, which is a small passageway that passes through the midbrain. Again, the fourth ventricle is the one that is continuous with the central canal and spinal cord. It contains several openings, which allows the cerebral spinal fluid in the ventricles to flow into the subarachnoid space. So again, all of these things are continuous. The first and second and third all flow into the fourth, and the fourth one is the one that ultimately connects to the spinal cord. And there you see, again here, see how they sort of look like ram's horns? Here, there's your horseshoe. But you see here's the little connection from the left and right that goes into the third ventricle, and then you have a connection that goes into the fourth ventricle, and then the fourth ventricle passes into the spinal cord. Cerebrospinal fluid is a clear, colorless liquid similar in composition to blood plasma. Its job is to protect the brain, to cushion the brain, to help maintain a constant temperature within the cranial cavity. It also plays a role in removing waste and increasing the buoyancy of the brain, and it keeps the brain from collapsing under its own weight. Keep in mind, the brain is a very soft organ, and it is easily damaged, and the cerebrospinal fluid plays a great role in protecting the brain from trauma. The choroid plexuses, I actually prefer the word plexi for the plural of plexus, but you can talk to your English teacher about why we sometimes say goose and other times we say gooses. Why do we sometimes say plexi for the plural of plexus, and why do sometimes we say the word plexuses? But that's enough of an English lesson for today. This is where the majority of the cerebrospinal fluid is manufactured. In each of the four ventricles, these are found in each of the four ventricles, where the blood vessels come in direct contact with the ependymal cells. The fenestrated capillaries are a particular type of blood vessel, and when you talk about blood and the venous system next semester, you will talk about the fenestrated capillaries. But fenestrated capillaries have gaps between the endothelial cells, which allow fluids and electrolytes to exit the blood plasma and enter the extracellular fluid. A fenestrated capillary allows things to diffuse that other capillaries do not. The choroid plexuses, holds about 150 milliliters of cerebrospinal fluid, which circulates through the brain and spinal cord. You have about 750 to 800 milliliters of that stuff produced every day. The old CSF must be removed as the choroid plexuses make new central spinal fluid. There are conditions where the process doesn't work and we have cerebrospinal fluid build up, and that can cause swelling within the brain. And one of the few ways, the old traditional way was simply, and now this is going to seem kind of gross, but to drill a hole in your head and let that that excess fluid leak out. The process of this production and removal occurs constantly. You will completely replace your cerebrospinal fluid every 5 to 6 hours. So about 4 times a day, there's complete turnover of the cerebrospinal fluid. The pathway from formation to circulation to reabsorption. The fluid and electrolytes leak out of the capillaries of choroid plexuses into the extracellular fluid of the ventricles. It's taken up into the ependymal cells. It is then secreted into vesicles as cerebrospinal fluid, circulates through and around the brain and spinal cord in the subarachnoid space. The movement is assisted by the cilia found on the ependymal cells. Some of the cerebrospinal fluid is then resorbed into the venous blood in the dural sinuses via the arachnoid granulation. And again, there is the cross-sectional artist rendering of how the cerebrospinal fluid works its way up and around into the subarachnoid space, and then back, and then down, and then to be recirculated once again. Here is the arachnoid granulations showing the cerebrospinal fluid going out into the extracellular space. The blood-brain barrier, we hear a lot about this. If you are a fan of the TV show House, Dr. House talks about the blood-brain barrier about every third or fourth episode they have. This is a protection for the brain. It separates the cerebrospinal fluid and the brain's extracellular fluid from chemicals and disease-causing organisms that are sometimes found in the blood plasma. Again, one of the things that is sometimes a problem with brain injuries and brain diseases is that typically medications can't pass the blood-brain barrier. Consists mainly of simple squamous epithelial cells and blood capillaries, their basal laminae, and astrocytes. Now, the unique feature of the endothelial cells in the barrier is that neighboring endothelial cells are bound together by tight junctions. And if you remember about tight junctions, they prevent fluids and molecules from passing between them, as their name would imply. This is influenced by the activity of the astrocytes on the developing brain. This limited capacity for movement into the brain and out of the brain by endocytosis and exocytosis. These many tight junctions really restrict the movement of things from one cell to the next, and from outside to inside. Substances that can easily pass through the plasma membrane and are able to cross the blood-brain barrier: water, makes sense. Oxygen, even more sense. Carbon dioxide, well, if oxygen can go across, then carbon dioxide's got to come back. And nonpolar lipid-based molecules. There are certain protein channels that will allow things that can't simply diffuse through: glucose, amino acids, certain ions. So again, those things make sense. Most large polar molecules are effectively preventing from crossing the blood-brain barrier in any significant amounts. And while the blood-brain barrier is a protection, it can also hinder access to medications into the brain, as I just mentioned. So here is a typical blood cell, typical capillary, with its openings which allow things to exit. So we've got water molecules and other small solutes that are able to very easily and readily exit that cell. Here we've got the astrocyte and its tight junction bound to that structure, and you see we now have these areas, like over here, you've got these big gaps where things can run out. Over here, those are all closed, and we have very limited ability of things to leave and enter the brain. Here's the thing about the blood-brain barrier. It is not a single structure. So you will never see a structure on a figure in a textbook labeled the blood-brain barrier. The blood-brain barrier isn't around the brain, it's within the brain. It's not located in any one distinct place, but it's found throughout the entire brain. The best way to think about this is you have to understand that the body's tiniest blood vessels, the capillaries, are designed to deliver oxygen and nutrients to cells and remove waste. So if we back up one, this is a typical capillary. And the way you see the opening, it is designed to let things leak out and leak in. See, they're not big enough for the red blood cell to come out, but they're big enough for all of this other stuff. And even larger, lipid containing things can exit those cells and also enter those cells. So you've got good stuff going out to the cells, and then everything the cell makes and produces waste can go back in to go the other way and be gotten rid of. But over here, let me go one more. Capillaries found in most organs and tissues are fairly leaky, as I just said. Allows for a wide variety of substances to move from the blood to the extracellular fluid and vice versa. Just said that. Capillaries in the brain, however, are specialized to allow only selected substances to enter the extracellular fluid. And so it effectively acts as a barrier, preventing other substances from doing so, from entering or exiting. The blood-brain barrier is a property of capillaries found throughout the brain rather than a distinct feature. So everybody take out your paper and your pencil and write down the blood-brain barrier is a property of the capillaries found throughout the brain, not a distinct physical barrier. It's not a membrane, it's a characteristic of the particular capillaries. Meningitis, infection of the meninges, is a potentially life-threatening infection, primarily found in the subarachnoid space. The inflammation has its classic signs of headache, lethargy, stiff neck, and fever. This is one of those things where people discover they have meninges because they have meningitis because they've had a dull headache for several days. Their neck is really stiff. They can't hardly stand to move their neck. They're running a fever. They're really tired and irritable. The only way this can be diagnosed is by examination of the cerebrospinal fluid for infectious agents and white blood cells. Bacteria and viruses are the most causative agents. Viral meningitis is generally mild and usually resolves itself within 1 or 2 weeks. Bacterial meningitis, on the other hand, can rapidly progress to the brain's involvement in death. Aggressive antibiotic treatment is necessary, and some of the most common forms are preventable with, again, the word they should not use the word vaccine right there. I don't like that word, but we will use it. All right, I'm going to stop right there, and then I will pick up with the spinal cord in a few minutes.